Hi, good morning, everyone. My name is Seitaro Terakura from Nagoya University. Um, and first of all, thank to organizers and Professor Natia for inviting me and giving me an opportunity to talk about our recent research. And the title of my talk is the Development of Eva1, a tumor-specific antigen targeting CAR T therapy. And I'll talk about insight from the development process, and I want to join the discussion after my talk. This is my COI. The Cured has offered the antibody sequences for the EVA1. The background is very, very simple. The chimeric antigen CAR T therapy is effective and already in clinical use. And all of you know the effectiveness. But the CAR T cell therapy for solid tumor has been extensively studied, but no CAR T therapy for the solid tumor has been approved so far. So everyone want to develop and with sufficient efficacy and clinical apply for the in near future. This is just the example of the development of the adaptive cell therapies in non-hematological solid tumors. And the bottom shows the CAR T therapy. And the target is including the PSCA, HER2, Claudin 18 here, and methylserine, and also MAC1. All of target antigen has been extensively studied, but no clinically approved one is existing. This is another example of the Claudin 6 specific CAR T cells, and recently published in the Nature Med in this year. The study is initiated from by the BioNTech company, and they are trying to amplifying the CAR response by combining the RNA vaccine in relapsed or refractory solid tumor. For those who are not familiar with Claudin 6, Claudin 6 is a very small protein located in the cell-to-cell tight junction, and the conformation is looks like here, and the 4 transmembrane protein. The blue ribbon is a transmembrane domain, and only small portion of the Claudin 6 is exposed to the extracellular domain, very small and very low antigen. And the data is in this study, 180 patients has been recruited into this clinical trial, and they tested the expression of the Claudin 6 here, and they look like these very, very dense patient has been selected, but actually 1/3 of the patients are densely Claudin 6 positive. And actually the germ cell tumor in the ovarian cancer cell and only small fraction of the gastric cell tumors, like 30%. Here's a 10%. Overall 30% are positive for the Claudin 6. And they started the cell processing, and then finally more than 20 patients are transfused by the Claudin 6 specific CAR T cells, and fraction of the patients are combined with RNA vaccines. Here's the waterfall plot to show the responses, and I think more than half of the patients exhibited the significant objective responses, but the more than 1/3 of the patients are showing the PR, more than PR. That is very, very significant responses if you think about the existing solid tumor CAR. But there's no sustaining CL cases. There's big response, more than 1/3 of the PR, but we did not see the CL cases. So the problem is again confirmed. We don't have many promising target antigen, and poor persistence in vivo is also the problem. So here's our target antigen, Eva1, epithelial V-like antigen. This is a single transmembrane proteins, very small, again, around 215 amino acid. And the conformation is here. This is extracellular topological domain. It's a very small and very low in the height. One of the immunoglobulin superfamily protein, and is known to be highly expressed in several solid tumors. And here's an example: the hepatocellular carcinoma and glioblastoma, small fraction of glioblastoma, and a large portion of lung cancer, showing the Eva1 positive. Also, the Eva1 is expressed under the downstream of the RAS mutation, a portion of the RAS mutated cancer showing the epithelial Eva1 positive. And also the Eva1 is involved in the activation of non-canonical NF-kappa beta pathway, and also activation of ERBB-PI3K AKT pathway has been shown. So Eva1 is associating in the process of the promoting the cancer cell invasion, proliferation, and metastasis. And also in the glioblastoma and the HCC, high Eva1 expression is associated with worse prognosis. But deficiency of the Eva1 in the homozygous deletion not lead to embryonic lethality. In this case, the genetic mutation is homozygous Eva1 are causing the hearing loss, but the person is not dead as an embryonic lethality. So we thought the Eva1 will be a good target for the CAR T cell therapy. So the purpose of this the study is the development of effective CAR T cells against solid tumor. And we did the three part of the experiments. First, we studied the confirmation of Eva1 expression in solid tumors. Here's the example of the tumor cell line. The pink is showing the positive. And we show the pancreatic cell, hepatocellular carcinoma, and cholangiocarcinoma, breast cancer, and lung cancer are all here positive for the Eva1 staining. But the ovarian cancer cell too are negative for the Eva1 expression. We also looked at the DepMap public data set, more than 1,000 cell lines, and here's the result. And mostly very high fraction of the tumor cells are highly positive for the Eva1, but some are not positive, like low-grade brain tumors like glioma, or the lymphoid malignancy, myeloid malignancy, and the melanoma is derived from the hematopoietic cells, are not expressing the Eva1. But if you look at the closely colorectal cancer and the lung cancer, non-small cell lung cancer, and the head and neck cancer, all highly positive for the Eva1 expression. Here's an immunohistochemistry example that we stained our own patients. And in the normal pancreas, Eva1 is not expressed, but in the pancreatic cancer cells here, positive, clearly on the membrane staining. This is Eva1 RNA expression on various normal organs, and ELO is EGFR, and both are almost very small fraction of the normal organs are expressing the Eva1. This is a lung and esophagus is a little bit expressed, the Eva1. So we conclude that Eva1 may be a good antigen for CAR T cell therapy against solid tumors. Next, we generated the CAR T cells targeting Eva1. We optimized 3 different portions as an antigen recognition site and spacer domain, and also intercellular domain. We optimized all 3 portion of the CAR T cells. And particularly CD79A, CD40 has been developed by the Dr. Jakrawale Juamane during her stay in Nagoya. And we have published about CD79A40 CAR can induce the sustained NF-kappa B phosphorylation. That's why the sustained response can be observed. For the optimization of antigen recognition site, we humanized the antigen antibody for the Eva1. And we used the humanized heavy and light chains created based on the structure of anti-mouse Eva1 antibody. And we prepared the 6 combination and tested the cytokine production and the proliferation upon the antigen stimulation. We did not see a significant difference in between these different antibody sequences, but the L282 has shown the most clear negative response for the Eva1 negative cell line. And also in the L282 sequence CAR showed the beta proliferation upon antigen stimulation. That's why we used the L282 for further experiments. We also tried to knockout cell knockout Eva1 in the NCIH1975 is Eva1 positive cell line, but we did the knockout and generate 2 different Eva1 knockout cell lines and tested to see whether the CAR T cells still respond to the these Eva1 knockout cells. And Eva1 can recognize the NCI original cell line, but the knockout cells cannot be induce any significant cytokine production. That will show that the Eva1 sequence CAR is very, very specific to the Eva1 molecule. Next, we try to optimize the intercellular domain, including the CD79A40, and comparing with CD28 or F1BB. And again, tested cytokine production and the proliferation, and we did not see any significant difference in the cytokine production. But in CD28, we did not see the significant proliferation, but 4-1BB and CD79A40 induced the beta proliferation. So we decided to do further experiment with these 2 different intercellular domains. Finally, we optimized the spacer domain. We actually did 5 different lengths of the spacer domain, but to shortcut the result, I will show you the short 15 amino acid and long 227 amino acid sequences. About the cytokine production, the longer spacer domain showed the higher interferon gamma production, but think about the proliferation. Shorter one shows the better proliferation. So the next one is the in vivo experiment to see the effect of the CAR T cells in mice model. Since we did not determine which is better for the longer spacer or the shorter spacer, we brought the 4 different constructs, long and short, and 4-1BB intercellular and 79A intercellular. 4 different constructs has been brought to the in vivo experiments. The target cell lines is NCIH1975 lung cancer cell lines. And on day -14, 2 weeks ago, 1 million cells are subcutaneously inoculated into the tumor, into the NOG mice. And then on day 0, 1 million CAR T cells are infused to the mice and see what happens. This is the individual tumor volume. And the red one is a longer spacer domain, and blue are shorter spacer domain. And we did not see a significant difference in between the intercellular domains, 4-1BB and 79A, but the longer spacer cannot derive the longer survival of the mice, but the shorter spacer derived almost all mice survived significantly. So we look at the peripheral blood expansion in between the 4-1BB short and 79A short, and we saw that significantly higher number of the CAR T cells are in the peripheral blood of the infused mice. There's another NOG mice model we tested. The target cells are CFPAC1 pancreatic cell. And also we put the 500,000 CFPAC1. Here's the IV model. And on day 0, 2 weeks later, we put the 2 million CAR T cells. Here's the only short spacer domain with 4-1BB or the CD79, CD40. And on day 0, we see a small BLI sign in the lung. So this may be the lung metastasis. But in the BLI sign, we see both CAR T cells can derive the significant survival, but a little bit higher, looks like beta 79A40 in the survival of mice. So here's a summary. We developed the CAR T for the Eva1 CAR T cells. CAR T cells specifically react to Eva1, and the antigen recognition site, intercellular domain, and the spacer domain length were optimized. In Eva1 CAR T, the combination of L2H2 short spacer, 4-1BB or CD79A40 was optimal. Eva1 CAR T demonstrated therapeutic efficacy against multiple cancer types in the carcinoma-bearing mouse model. So why the longer spacer domain can derive the better result? Last year, the Science Immunology paper says tested the proximal epitope and distal epitope. And if you use the proximal epitope, the CAR can recognize the proximal epitope. That's why the T cell and the target cells makes more closer T cell synapse. But if you use the distal epitope, the CAR only cannot make the closer synapse like this. That's why the CD45 cannot exclude in this situation, but in the proximal epitope or the shorter spacer domain will make this close T cell synapse that will exclude CD45 from this T cell synapse. So as a CD45 is a dephosphorylating the LCK. So if the T cells can make the synapse and activate it, the LCK will phosphorylate it and help more sustained T cell activation. But CD45 has to come and dephosphorylate the LCK to deactivate the T cells to maintain the situation. But if you use the shorter spacer or the smaller antigen, because Eva1 itself is very small, the then CAR recognized site of the Eva1 is also proximal to the plasma membrane. That's why Eva1 CAR T cells supposed to be activated with a signaling blockade or the dephosphorylation of the CD45. If you think about other target antigen, here's the existing CAR, BCMA, CD19 are the only small, very, very small extracellular domain, and the height is very small. But if you think about other target antigens like B7H3, GPC3, HER2, MAC1, all are coming from the target of the antibody therapy. So they are using the same antibody with the antibody therapy. That's why the target molecule is very big and the height is high. Probably that's why we see sometimes very weak or the bad proliferation upon this stimulation. That's what I'm thinking. So here's a future prospect. Because Eva1 is expressed in the lung and the esophagus organ, a very small fraction, but we think about it. So to determine whether the Eva1 CAR-T can recognize the Eva1 and the normal organs, we are currently developing the all-mouse Eva1 CAR-T to see if the Eva1 can recognize the normal organs. If all mouse CAR-T experiments show no major problem in mice, viral vectors will be created for the human clinical trials. Yes, that's all. I will stop my talk here. And here's the acknowledgement. The Eva1 CAR has been developed and tested by Masahide Osaki. And as I already talked in the last inaugural session of GTCI, I am thinking about combining with CAL5 knockout with Eva1 CAR T cells. And also Jakurawade Juramani is tested the CD79 and 40 and cured people as offered the antibody sequence. Thank you. Thank you for your attention.